The origin of species.
Just out of graduate school, zoologist Ernst Mayr did not have any field experience. That was not a problem though, because he had three things going for him, which were a lifelong love of birds, tremendous enthusiasm, and the financial help of the British banker and amateur naturalist Lord Walter Rothschild. While Mayr had many downfalls such as bad weather, treacherous paths, and repeated illnesses, he ventured to New Guinea on a one-man expedition in which he obtained 26 species of birds and 38 species of orchids. With all of his great success, Mayr wrote quite a few books. One in particular is titled Animal Species and Evolution, in which Mayr revealed the fact that the natives of New Guinea recognized 136 different types of local birds. Similarly, Western zoologists, using traditional methods of taxonomy, recognized 137 species. In other words, both locals and scientists had distinguished the very same species of birds living in the wild. This concordance between two cultural groups with very different backgrounds proved that the discontinuities of nature are not arbitrary, but an objective fact. Perhaps the most striking fact about nature is that it is discontinuous. When you look at animals and plants, each individual almost always falls into one of many discrete groups. These discrete groups are known as species. But before we can understand the origin of species, we need to figure out exactly what they represent. One answer is based on how we recognize species, as a group of individuals that resemble each other more than they resemble members of other groups. For example, tiger would be defined as the group including all Asian cats whose adults are more than 5 feet long and have vertical black stripes on an orange body, with white patches around the eyes and mouth. We do see similarities between groups that coexist in the same location and yet never exchange genes. The members of one species simply don't hybridize with members of another. The groups are therefore reproductively isolated from one another, which means they constitute distinct gene pools that don't intermingle. Thus, species are distinct not merely because they look different, but because there are barriers between them that prevent interbreeding. So reproductive barriers may keep members of two related species from mating with each other. Species might not interbreed simply because their mating or flowering seasons don't overlap, the species are somewhat isolated by habitat, or they may also differ in courtship displays, so even when they do meet, members of the two species rarely mate. Finally, when they do succeed at mating, the sperm of one species is poor at fertilizing the eggs of the other, so that relatively few offspring are produced, and half of these hybrid offspring, all of the males, are sterile. Therefore, putting all these barriers together, we conclude that the species exchange virtually no genes in nature, and this result has been confirmed by sequencing their DNA. These can be considered good biological species. According to the problems, the biological species concept is still the one that evolutionists prefer when studying speciation. This is because it gets to the heart of the evolutionary question. Basically, under the biological species concept, if you can explain how reproductive barriers evolve, you've explained the origin of species. Another question. How do you get one species to divide into two, separated by reproductive barriers? These species are known as sister species, which are species that are each other's closest relatives, that were often separated in nature by geographical barriers. Mayer argued that these barriers were merely the byproducts of natural or sexual selection that caused geographically isolated populations to evolve in different directions. These form, indirect methods must be used. This includes testing predictions derived from the theory of geographic speciation. This method tells us that it takes approximately between 100,000 and 5 million years to evolve two reproductively isolated descendants. It is important to understand that geographic isolation is common and can occur from many different things, such as mountain ranges rising, glaciers spreading, deserts forming, continents drifting, and droughts that divide. Each time this happens, there is a chance for a species to be split into two or more populations.
So there are three different theories of geographic speciation, and this slide is going to go over the first theory. It says that if speciation depends largely on isolation, then the populations must have undergone many opportunities for isolation. This includes geographic isolation, long-distance dispersal, and reproductive isolation. So when it says that speciation depends largely on isolation, then the populations must have undergone many opportunities. It means that since there are many opportunities, therefore that is what is being let out, which is the dependence of speciation on isolation. The next theory of geographic speciation is that if populations must be physically isolated from one another, then the most recently formed species should be found in different close-by areas. This necessarily doesn't mean that they're going to be very close by, as in one region of China to the next, but maybe one area in Asia connected to one place in North America that may have been connected previously from Pangaea or the Bering Strait Bridge, somewhere along those lines. And we can tell by looking at DNA in sister species and working backwards. Many times, the different species are found in separate areas. So Hawaii's Drosophila flies have the oldest species on the oldest islands and the newest species on the newest islands. So we compare chromosomes and DNA, and from there we work on to see where each species falls within a timeline and within the geographic location. The last theory states that reproductive isolation between species that have been physically isolated increases slowly with time. So the author of the book, Jerry Coyne, and his colleague Alan Orr found that Drosophila flies in Hawaii have a gap of 2.7 million years in which genetic barriers have become strong enough to prevent interbreeding. So what can we say about all of these theories? Well, they all hold water. They all have evidence to prove why it's a possibility and known. Isolated populations do diverge, and reproductive barriers develop as a byproduct of evolution. This is all proven by these three theories. Are there any other theories besides geographic speciation? Well, of course there are. It's called sympatric speciation. And this is the theory that we have divergence without geographic isolation, meaning we have a difference in speciation without necessarily having them in different parts of the world or country or so on and so forth. So speciation can occur without geographic barriers. Is sympatric speciation possible? Of course it is. Sympatric speciation is possible under restrictive conditions that are uncommon in nature, but it doesn't make it impossible. As mentioned before, diverging members may interbreed and cause a genetic or generic single species, meaning that they could hybridize and it's just one species living in the population rather than many. That is the probable idea. How can we find proof for sympatric speciation? Well, two species that are similar living close by are not necessarily originally from that area, as species do tend to move around. So that can't be our proof. Rather, proof is found in places like habitat islands that are too small to have geographic barriers. If there are related species, this may be a form of sympatric speciation. It doesn't necessarily prove that it is sympatric speciation, but it could be a form. The example that we have are the cichlid fish that habitate in two tiny lakes in Cameroon. These lakes are basically filling up volcanic craters in the country, and they are separated by 0.2 and 1.6 square miles. And these two lakes actually have 9 different species in one and 11 in the other. And you can definitely tell the difference in the species if you look at these pictures. But they do diverge from one common ancestor. Another present example of sympatric speciation is the palm trees on Lord Howe, an island in Australia or off the coast of Australia. It's very small for an island. It's about 5 square miles, but it contains 2 palm trees that are closely related. The Kentia palm, which is a houseplant throughout the world, and the curly palm. And they're actually very close relatively, but another example of sympatric speciation. This leads to the question if sympatric speciation is common. Well, it is given many opportunities to occur, but given these opportunities, it doesn't occur very much at all, not very often, and it's very difficult to find examples of sympatric speciation. So, although it is not a common occurrence in nature, it can actually be found in plants, and there are 2 types of sympatric speciation that are common in plants. Allopolyploid speciation is one of them. It starts with the hybridization of 2 species that live in the same area. They must have different types and numbers of chromosomes in order to work, and they form from doubled up chromosomes from 2 parent plants. Then we also have the autopolyploid speciation, which form from simply doubling the chromosomes of 1 species instead of 2 hybrid species. So it actually forms fertile hybrids when mating with one another and sterile hybrids from mating with parental species. Either way, whether we're doing either form of ploid speciation, we need the formation and union of sperm and egg, which plants have, and abnormally high numbers of chromosomes, which are more than possible in plants. About a quarter of all flowering plants actually have been formed by some type of polyploidy. This leads to the question of, does polyploidy occur in animals? Rarely, more so, it occurs in fish, insects, worms, and reptiles. We really don't see it much in mammals. And the polyploidy occurs mostly asexually in these animals. One mammal actually does undergo polyploidy, and it's the red viscacha rat of Argentina. It has 112 chromosomes, so it fits the criteria of having the high number of chromosomes that are necessary for polyploidy speciation. So why is polyploidy so rare then, especially in animals? Well, it disrupts the XY chromosome formation that we have. Basically, asexual reproduction leads to the chromosomes that make up whether it is male or female. There is a lack of self-fertilization in animals. Animals typically fertilize together. They have sexual reproduction, while plants can do self-fertilization. It involves changes in chromosomes, so the doubling up and the hybridization. And it is much faster than geographic speciation, as polyploidy can occur in a mere 2 generations, while otherwise it takes a much longer time. Now, the evidence for polyploidy is rare, but it has been proven in other ways. It has occurred multiple and countless times over the course of evolution, and we can prove this because it has actually been synthesized in laboratories, and it provides proof of evolution, so it backs it up. Darwin would actually be proud, according to the author of the book, if he had seen all the findings that we have found.